So the next session, I would like, it's one of my great opportunity to introduce the next speaker, Seth. At this moment, he is a business development manager in life science, Southeast Asia. He joins Avantor Performance Materials as an assistant manager, technical applications in 2018, and based his work based in Malaysia, and he's responsible for creating new business opportunities, and especially in life science business in Southeast Asia, including Taiwan. And before joining the company, he gains lots of experiences, more than 15 years experiences in life science research, including technical sales, service engineer, and business development. And of course, he completed his doctoral degree from Malaysia, University Putra Malaysia in 2003. And it's my honor to introduce him, and he will give a talk entitled The Impact of Serum Quality Towards the Performance Consistency in Cell Culture. Please welcome Dr. Seth.
ค่ะขออนุญาตนะคะหลังจากพักเบรกมาขอบคุณค่ะอาจารย์ที่แนะนำแทนนะคะขออนุญาตแนะนำตัวนิดๆนะคะตัวเวลานิดนึงชื่อธรัชยาวรักติวงศ์นะคะเป็น Business Development Manager ของบริษัท Avantor แล้วก็ VWR ในเมืองไทยนะคะวันนี้ก็คือได้รับเกียรติจากทาง INNOLAB ในการเชิญมิสเตอร์เซธที่เป็น Business Development ของเราในกลุ่มไลฟ์สไตล์ที่มาจากมาเลเซียแล้วก็สิงคโปร์นะคะมาแนะนำเกี่ยวกับว่า VWR ไลฟ์สไตล์ทั้งหมดมันมีอะไรบ้างแต่เนื่องจากว่างานนี้เป็นงานกึ่งวิชาการเราก็เลยไม่ได้อยากจะแบบว่าอินดิเทลเรื่องของการขายของเยอะนะคะคุณเซธเนี่ยเขาวันนี้เราจะบอกเขาว่าให้เขาสวมหมวกเป็นสปีกเกอร์จริงๆในเรื่องของงานโมเลกุลาร์ไลฟ์สไตล์เซลล์คอลเจอร์ทั้งหมดโดยจะไม่ค่อยได้แตะผลิตภัณฑ์เยอะแต่ว่าเนื่องจากมางานนี้แล้วอะค่ะก็ถือโอกาสนิดนึงด้านหลังก็จะมีบูธของบริษัทนะคะหลังจากเสร็จงานพักเบรกอาจจะแวะก็มีเยี่ยมชมนะคะเพราะว่าวันนี้เรามีของแจกเยอะมากมีพวกฟิโลโลจีพิเศษมีพวกเซนซิทิวทิวต่างๆนะคะก็อยากให้ทุกท่านขนกลับบ้านไปให้หมดเลยนะคะเราแจกหมดทั้งงานเลยก่อนที่จะแล้วก็มีถุงด้วยเผื่อจะไปเดินตลาดต่อตอนเย็นนะคะเพื่อไม่ให้การเสียเวลาก็เซฟโอเคแล้วค่ะขอบคุณค่ะ
Thank you, everybody. Now it's close to noon time, so good morning and good afternoon. So my name is Seth. So yeah, very nice introduction, and of course I wish to introduce myself a little bit in less formal way. So yeah, so this who am I? So I was a doctor of veterinary. So yeah, I spent 5 years working with animals. Try to find a solution how to treat them or all these things before I go to actually do 6 years in cancer research. Okay, and just now during break, yeah, I talked to them that I was a service engineer before for the molecular biology instrument. All right, so yeah, this is where am I in Switzerland undergoing the training, having some good time, wearing jeans, all these things to try to repair the instrument. Okay, and of course. Then after that, I have a first commercial role as a product manager. All right, also supporting on the cell culture and also molecular biology. During that time, I have a very good opportunity, like working with certain very hot topic, like for instance the stem cell, CAR-T, CRISPR, obviously. So that is who am I. So today, before I go to the really a topic, I wish to introduce to you all a little bit on our company, all right? Because I was really fortunate that I joined at the time of very big merge, which is Avantor and VWR, all right? So in the year 2017, VWR acquired, Avantor acquired VWR, all right? So this is a very good marriage because why? Because Avantor is actually a chemical company. They have lots of chemicals, like including the cell culture chemicals, CGMP produced. And whereas VWR is actually a global hypermarket for the life science product as well. Alright? So with this merger, now this is our new logo, Avantor. And of course, for those that you are not very familiar with our company, so we have a presence in the Southeast Asia, across Southeast Asia and Taiwan, right? So me personally, actually, I'm based in Malaysia, all right, and then I support the entire Southeast Asia and Taiwan, right? Okay, so this is what, just a little bit on the product introduction. So this is what we have for the molecular biology, starting from the sample prep until the cloning, sequencing, and plotting, all right? And also on the real-time PCR as well. So because of the VWR branch, so we are actually the distributor for certain brand as well, right? So for the real-time PCR, we are actually distributing on QuantaBio. And also, we do have a protein analysis, or what we call Western blot workflow as well, all right? In other words, we are actually have a complete solution for the life science. Yeah. So now is actually finished my talk on the commercial side or on the introduction of the company. So we will actually go to the real topic for today. So today, because of the serum, so I wish to introduce my key player here, which is the cow, where we are actually getting the serum from, alright? So a lot of majority of the serum that is actually we use in the cell culture today is actually from cow, alright? Then it's depending on the age of the cow, then you can actually classify it as the fetal bovine serum, newborn calf serum, or calf serum, alright? So it depends on the age. So after collecting the blood here, get it clotted, and then collect the serum, and then this is the end product that you will be using in the cell culture as the supplement, alright? So today, the topic I will start with some simple basic of what this serum is all about and why it needs to be used in cell culture. And because when we do cell culture, it's actually we are taking the cell from the body and letting it grow on the petri dish. In other words, it's from something in vivo to become in vitro, right? So in order for the cell to grow, we need to supply a lot of nutrients. So that's where the medias and serum comes from, right? And of course, if you are doing actually something that is a small scale, using the cell culture flask, then the consistency of the serum is not a very big issue. But what if you translate everything into a very large scale, like for instance, bioproduction, which we will be using for recombinant protein, like for instance, EPO that is used for the therapeutic, or the large amount of the cell for the cell therapy. So we need to have a very good quality of the serum to be used, right? And of course, what is the criteria you need to, as an end user, what is the criteria you need to look at when you want to purchase a serum, all right? And of course, in order to do all these things, of course, we have one publication to show how actually we can test the serum quality, right? So, what is serum? So how is serum is collected? So actually serum is collected from the animals, alright? And it is done just like we doing the blood donation, for instance. And then we let the blood clot from the animal, from the abattoir, and then keep it at a very low temperature, alright? And then before we do the testing, we bottle it, and then before you actually receive it in a bottle format, alright? When you get the serum, besides you getting the bottles of the serums, you're getting a lot of documents to show the quality of the serum as well. What is like for instance, what are the tests that we are using to prove that it is actually of very good quality of the serum, which I will discuss on this. Alright, why serum needs to be used, or why serum is widely used in the cell culture? Alright, in order for the serum to be used in the cell culture, it needs to have a very low antibody level. Alright, that's why it is better that, for instance, as young the age of the animal, it will be the better. Because during that time, the animal doesn't really expose to the pathogens, so that the antibody level is actually lowest. Alright, and of course, we need to have a very high content of the growth factor in order for the cell to grow better. Alright? The most important supplement from the serum is actually the growth factor for the cell to grow. Alright? And of course, serum does serve as the carrier for the minerals, fatty acids, ions, hormone, or even acts as a hormone itself, alright, to the cell to grow. And of course, it contains the protein that enhance the attachment. Because cell, there's two ways for the cell to grow in the outside the body, in the cell culture flask. It could be remain as the suspension cell, floating, or it needs to adhere to the cell culture flask, alright, attachment. It depends on where is the source of the cell is being gathered from the tissue. Like for instance, if the cell is actually getting from the liver, there's a tube from a tissue to become a cell in the cell culture flask, most likely it will actually attach at the cell culture flask. Alright, if the cell is actually taken from the blood, which is by nature is actually floating, so most likely it will become a suspension cell in the cell culture flask as well. So one of the main reasons why serum is being used is also for the attachment as well. So later on I will discuss a little bit on the serum-free media, where a lot of customers is actually complain why my cell doesn't attach where it's supposed to be attached. Because there is no serum, so that's why we need to add in certain attachment factor for the cell to attach to the cell culture flask, right? So in the lab, what is the step that actually cell culture people or technician is doing? It's actually every day they need to actually take out or remove the used media and supply with the new media. Or even they need to actually do the subculture or dividing of the cell from one flask to another flask. Because we need to get the cell to grow actually. Alright, and then all this is being done in a cell culture incubator, CO2 incubator, alright, and grow by about 37 degrees. And on and off, we need to actually observe the morphology of the cell under the microscope as well. Alright, and of course, this is actually quite elaborate step for the cell to grow outside the body. Alright, and then as you can see that the process is quite laborious. Why the customer wants or why you all want to use cell in the lab? Because cell can be served as the disease model. Alright? So because in order for us to understand more about the disease, a lot of time we actually start from the cell. Alright? We understand how is the cell respond to the treatment before we actually test it out in the animal as well, or even the human. Alright? Or cell as a factory, which is like the cell, you are using the cell as a factory to produce the recombinant protein that you want, like recombinant equal. Alright? Or cell as a therapy, like for instance, you are using the cell for the cell therapy, which is in the stem cell research now. Alright? So these are the three main applications why people are still using cell culture now. So, in order for the cell to grow outside the body, alright, we need to supply all the nutrients for the cell. Alright? So normally we were using something called basal media. There's a lot of type of the basal media in the market. Like for instance, RPMI, DMAMs, all these things, and the list is actually very long. And actually in the basal media, it contains all the chemicals that is needed or all the nutrients that is needed except the serum that is needed for the cell to grow. And when we need to actually add in the serum to make the media complete. So this is what we call a complete media. Alright? And certain customer or end user love to add in the antibiotic and antimycotic in order to prevent the cell culture from contamination. Alright? And sometimes we do encounter that certain customer is actually add on more amino acids or vitamin in order to grow the cell better or produce more recombinant protein that they wish, for instance. Alright? So this is more on the modification side. So let's go back to here. So here in cell culture flask, we are actually easily talking about 10 ml, 5 ml, or even 25 ml, or even 50 ml of the media. Alright? So at this point of time, actually customer is already very concerned about the quality of the serum already. Not to mention that when they actually upscale it to a bioreactor, where we are actually talking about 100 up to 500 liters or even 1,000 liters of the bioreactors, where we need to have a huge amount of the cell growing in the bioreactor in order to produce the, for instance, the recombinant protein that you want, or produce the cell that will be used for the cell therapy. All right. So at this point, the consistency of the cell to grow is really reliant on the serum that you are actually using. If you are still using this serum supplemented media instead of serum-free media, all right? So, across the years that I'm supporting the cell culture customer, these are a few issues that the customer is actually facing, all right? They will actually tells me why you deliver 20 bottles of the serum, but there are some bottles of the serum is actually more red in color, some is actually more normal looks like the colors. So the customer is actually facing on the color inconsistency. Sometimes they do receive certain bottles of the color is not clear, or there could be. They are very concerned or not whether or not the serum is actually contaminated, right? And some customer will complain to me that why the media or why the serum there is something that's floating inside. Is it a contamination? All right. So these are a lot of customers will actually use a physical observation to actually tells you, okay, your serum is not good or your serum is better. All right. And of course, now a lot of customers has been educated that it is very important for them to know that the traceability of the serum. For instance, in the cell culture world, in the fetal Guan serum, there is a lot of countries that actually origin of the serum. For instance, serum can come from USA, North America, South America, Australia, New Zealand, for instance. So, especially for the North America, the price of the serum is actually very expensive. So, and this is also one of the gold standard for the bioprocess customer, because they need to know, if you're telling me that the serum is actually from North America, are you sure that it is actually from North America animals? All right. So there's a lot of documents that you need to actually check in order to ensure that whatever is written in the label is indicating it is North America serum is actually indeed come from North America. All right. And this is very important. Like for instance, for the bioprocess customer, certain customer even goes to the next level. They wants to have the serum from Australia or New Zealand. One of the main reasons is that they do not have a mad cow disease there, so this is easily for them to actually to produce a safer biosimilar product, for instance. Right? Next to traceability, the customer is very concerned to lot consistency. All right? This is very critical as well, because like if the consistency is not consistent, so now if the customer is purchasing this lot and compared to the previous lot, maybe the cell growth might be different. So this will affect a lot especially in the bioproduction, because all this is actually very linearly increased as the volume of the bioreactor is increased. Right? So a lot of customers will really look for the source that has as close as possible the lot-to-lot consistency. Okay, but to make things worse, certain customer purchase from a vendor that after they test 3 or 4 lot and then identify the desired lot to reserve for the entire years, when they receive the serum, they might check the performance of bottle per bottle in a lot. All right, they might still find out that certain supplier might produce the serum that is like not so consistent bottle to bottle. So again, they will feel that, wow, why should I spend so much time to actually test each and every lot before I identify the lot? And then now when I receive the lot that I desired, but yet bottle to bottle within the lot is inconsistent. All right, so this is actually will throw a lot of time wasted. All right, and what about ink trend? If the serum suffers a lot of bottle to bottle consistency, which means that from one bottle to the next, the nutrient level is actually different. Alright, and then the cell culture, the cell will grow better in this bottle versus the other bottles. Right? And of course, customer will also check on the documents to check on the endotoxin level, because when we actually get the serum from the animals, we want to make sure that there's the minimal contamination as possible, if it is no contamination at all, in order to prevent that the antitoxin from the contaminants is actually passed through the filtration. Because endotoxin you can't actually filter from the filtration, so we need to ensure that the source of the serum is in a very good quality, and the animals used in the serum collection is actually of a very healthy animals, to ensure that the endotoxin level is actually as low as possible. Alright? And also another important parameter is the hemoglobin level. This is actually indicates how well is the blood is being collected at the abattoir. Alright? With all this inconsistency in mind, okay, with all this inconsistency in mind, so now there's a lot of document that needs to be supplied by the serum supplier, okay, and in each and every document, it will tell you what is the test that is been done in the serum, for the serum before they get to your, right? Just now I mentioned about the traceability. So if you purchase a very high quality or very high price serum from USA, because you want to have a very good quality of the serum from the country that do not have certain diseases of importance, especially like mad cow disease. So you want to make sure that in the bottle, the serum is indeed comes from USA, from the animals that is collected in the USA, grown in USA, and everything is processed in USA as well. Right? So there's one organization called ISIA. It will do all this checking or auditing to ensure that whatever serum that is registered under their organization, something like ISO, alright, is actually indeed starting from the abattoir until the bottle, logistic, everything is actually done properly to ensure that there is no added of the serum from other country. Okay, so in other words, it will ensure that based on the document and check-in and auditing, you are actually receiving the serum 100% from the USA. Alright? However, some customer is actually questioned about this because all this is just documentation, just auditing. If there is no one that is actually proving that there is presence of the non-USA serum in the bottle, of course this company will always receive always get this certification, right? Because no one is complained. So in the auditing, everything document is supporting it is USA, so everything goes well. So that's why recently there's another company called Origin. This actually helps the serum provider to actually to do a fingerprinting to ensure that the serum that is in the bottle is actually from the USA. What does it do is based on the very simple way of doing it is, I will give you an example. If a cow is actually grown in US, this cow will actually breathe the air in the USA, eat the grass in the USA, drink the water in the USA, step on the soil in the USA, right? And they managed to find out that all the serum, right, there is a certain radioisotope that is present in the serum to indicate all this is actually from USA. And it is actually quite different from Australia, South America, for instance. So based on this fingerprinting, then you can actually ensure that the serum that you get is actually from USA and not from other countries. Okay? So this is an additional test that can be done by certain country or certain vendor of the serum to ensure that actually what they claims is correct. Alright? And I wish to draw to you another good thing about it is cold chain manufacturing. So this is not something that is usually been done by the serum supplier. Okay, what does it mean is when the serum is being collected from the abattoir, of course it will actually keep it in the room frozen until they receive the enough volume for a certain lot. Alright? Then a lot of company is actually let it thaw and let it process and bottle as soon as possible at a temperature that is maybe close to ambient temperature. Alright? So as you imagine that serum contain a lot of growth factor and a lot of protein that is undefined as well until today. And actually a lot of them is actually at high heat sensitive. So if you are processing the serum in high temperature, so which means that you actually subject all these heat sensitive protein to the degradation. Alright? That's why one of the effort that is done by our company Serum is actually to reduce the manufacturing temperature, especially at the filtration process, as close to 5 for the whole entire process, the sample of the serum doesn't actually increase more than 5 degrees. In other words, we are not only control the temperature of the environment, we actually ensure that the serum is actually never been processed at a temperature more than 5 degrees. In other words, we managed to actually ensure that there's more nutrition, or as close as possible, the serum that you get is actually at the same nutrition level as what the cow is actually getting when they are from their blood. Alright? And this is actually very important because it will actually ensure that your cell is actually getting more nutrient to grow. Alright? And of course, other things that has been done across the industry is actually we will actually using a single-use filtration technologies. In other words, this will actually reduce the cross-contamination. If you actually reuse the filtration, so which means that you are subject to cross-contamination. And of course, in order to get rid of the pathogens contamination when we are processing the serum, actually we will actually use the triple 0.1 micron filtration. Alright? And of course, we actually subject all our serum into the ISIA regulation, as well as also sending out the serum for the origin verification. Alright? So with that, you will actually get a very good quality of the serum that is clear, alright, consistent in the colors, consistent in the quality, and consistent in the bottle to bottle within the lot. Alright? This is the consistency in the bottle to bottle is actually achieved by the cold chain manufacturing as well. Alright, you just imagine that if the serum is actually been produced or been processed in low temperature, which means that the company is actually allowed, or the manufacturing site is allowing the serum to be in the manufacturing site for a longer period of time, where it gets to stir better, filter more better. Alright? So in other words, you will get a more homogenized bottle to bottle within the lot. Alright? So this is something that's very important. In order to prove that the serum is actually of good quality, actually we do something called low density plating efficiency testing. Alright? To compare it with the serum of the same grade on paper from other manufacturing. Alright? So what does it mean by the low density plating is we are actually growing the cell in a 96 well plate. Alright? At the low cell shading density as possible. Because a lot of cells, they cannot grow alone. They need to grow with their colleagues together. Alright? So because if you are putting the cell alone in a single well, most likely the cell will die without detection. Alright? So we will actually check what is the seeding density that is minimal, but the cell is still survive. Alright? So that is what we call a low density plating efficiency testing. And this is actually a very stressful test for the cell. So in order for the cell to grow, you need to have a very good serum quality. Alright? So this is why we choose this cell. These are actually this test to do it. So we are actually choosing 2 lots of the serum from the company. All right, all are US origin, and we send the same cell to the testing lab using their control serum to ensure that the result is actually reproducible. All right, and also we compare with the serum from the other company as well, 2 lots as well each. All right, and also during the testing we also test something called FB essence. So FB essence is something that contain lower concentration of the serum, but we increase certain, we add in certain growth factor and some probability information of the growth factor into the mixture to ensure that the cell will grow as you are supplying 100% of the serum into the supplement. So why we actually develop this product is actually to ensure that the cost of the bottle of the serum is actually reduced, but the performance doesn't get compromised. All right. So this is what we call FB essence. All right. And during the test, we also reduce the serum concentration. Okay. Normally, most of the cell culture lab you go to, the customer will tell you that, or the researcher will tell you that it is actually running at 10 degrees 10% supplementation. All right, and you ask them why, they will say, yeah, this is a standard SOP, right? But do we actually really need to have 10% of the serum in the complete media? This is something that the customer is always asking themselves, but a lot of time they still stick to 10%. All right, and as you know, the serum price is getting increased every year. So if there is a chance for you to reduce the serum concentration, you will save a lot of money in your lab. All right. Not to mention that if you are actually doing the bioprocess, serum is actually contaminants to the recombinant protein that you are produce. All right. So in terms of the purification burdens, you can actually reduce the burdens by reducing the serum as well. All right. In other words, the cost for you to do the purification is much more cheaper. So in other words, you can actually reduce the overall cost of operation by reducing the serum concentration and also the purifications. Right? So what we do is we reduce it from 10 to 7.5 to 5 and 2.5. Okay? And everything is done in the 96 droplet. And we are actually checking the colony of the cell after the culture, after 10 days of culture. All right. So in order to avoid human error and bias, we are actually using automated spot counting to ensure that it is actually more objective in terms of the counting. All right. And don't worry about subjecting the cell to grow in the 96 droplet for 10 days, because we are actually using the media containing a stable L-glutamate, which doesn't actually easily change to ammonia, which is toxic to the cell. So in other words, we are actually providing the optimum condition for the cell to grow, and whether or not the cell is grow more happy or not, it all depends on the serum performance. All right? So this is, sorry, I think I hidden one slide. No worry. I can actually show you from here. Maybe I can unhide that slide first. Okay, so this is one of the cell line that we are using. Actually, we are using 3 cells. Okay, one of them is actually MBCA, which is actually the canine cancer cell. It resembles the cell line. Another one cell is WI53, which is more on the primary cell line, and the other one is CHO cell, which is mainly used for the bioproduction. Right? So which, as you can see that in the graph, TA, TA. So which means that T8 and T2 is a replicate of the 2 lots from Ceradyne filter bovine serum. T3 is actually the control lab that is doing for to double check on the result. T4 is actually the FB essence, which I mentioned to you, the serum's alternative. T5, TF5, and 6 is actually competitor product from one company, and T7 and 8 is another company as well. Alright? And as you can see the bar chart, 10, 75, red color is 10, 75, 7.5 is red, black, 5 is actually green, and 2.5 is actually pink, purple. So from here, you will notice that, okay, it's not necessary for you to actually increase until 10% of the serum, alright? Because you will actually, sometimes you will find that certain cell, especially the cell you might be using, will grow better at lower serum concentration. This is what we've shown here, alright? And of course, as you can see that certain manufacturing, the performance is actually much more lower, although you are actually supplying the same concentration of the serum, alright? Because why? Because actually Ceradyne serum is actually produced in the cold chain temperature, so we manage to preserve much more of the nutrient to ensure that the cell will grow better, alright? And even in our serum supplement, we observe the same cell morphology or same growth morphology, even the component doesn't contain 100% of the serum. It's actually reduced serum concentration. Alright? So the same observation is actually observed across 3 types of cells. Alright? In summary, actually serum from the Ceradyne actually provide a very consistent superior nutrition support, alright, compared to competitor brand. And it is actually not only for one lot, it is actually more than one lot. Both lot is actually produce a very superior result, which means that lot to lot variation is actually reduced. Alright, and also we wish to highlight to you that the FB essence, which is the serum alternative, will have having the same performance or even better performance than the pure 100% FBS. Right? So I wish to draw you some information from the COA whenever you buy any serum for any company. Alright? So of course it will tell you that beside triple filtration, there maybe there's certain company will give you like certain additional treatment, like for instance gamma irradiation, so on and so, or heat activation, depends on the customer needs. Alright? And then they will tell you that actually when is the manufacturing started for this batch of the serum, and when is the expiry date. Normally the serum can last for about 5 years expiry date. Alright? And how is the cell is being how is the cell is being filtered? Like for our case, it's actually filtered in 0.1 micron sterile filter, 3 times, and all the blood is actually collected and processed in USA. All this is being mentioned because we need to adhere to the ISIA standard. Alright? And one thing I wish to draw your attention is the total volume of the lot. So if you check on the across different manufacturing, it's good that you can ask what is the volume for the lot, each lot, so that if you test a certain lot that you are giving you the desired quality, then you can actually preserve the whole lot. This is very important, especially if you are doing bioproduction, because if the lot volume is actually very low, then which means that each year you need to repeatedly check the serum quality as well. Alright? And of course, there's a lot of testing on the important diseases, virus diseases. Alright? So in our case, we're actually using qPCR to do the testing. Alright? And here, because we already have an additional test, which is the origin, to ensure that the serum is indeed is from USA, so it will tell you that whether it's pass or fail. All right. And here, we even goes to check on the hormone profile of the serum as well, right? And in the statement, we will make sure that we highlight that we are actually the ISIA certified organization since the year 2013, and also we have an additional origin verification test. And in our test, we will actually ensure that all our animals is actually collected from the facility that is already certified do not contain BSE contamination. And beside ISIA, certain bioprocess customer also indeed request whether or not we do have the European qualification as well. All right, so this is additional qualification that we have, which is from the European. Okay, so this is how we can actually check the quality of the serum before you actually use it in the research, and how important is the consistency of the serum quality towards your cell culture. And before I end, I wish to throw to you like a few minutes, because across the years, customer will face a lot of problem in testing of the serum, and yet the result is not consistent because there's a lot of lot-to-lot consistency and bottle-to-bottle inconsistency issue. So now there's actually a trend for the customer to actually go to serum-free media or serum supplement, which is instead of using a component from the animals, actually all these supplement is actually chemical defined and added with some recombinant protein. So every single component is well defined. All right, instead of having a lot of undefined material inside the serum. So it will give you a more lot-to-lot consistency, right? So because of that, we just get the authorized regional distributorship from a serum manufacturer in Germany called Avantor, and they have a lot of the serum-free and serum supplement product for you to choose based on the cell line that you are using. All right. With this, thanks a lot, and yeah, this is my contact, and feel free to contact me if you have any question about the cell culture and also the serum. All right, thank you.
Thank you very much.